All right, without further ado, Alef at College Rising Junior at the College of Worcester. Alef, come on up. Look it up.
Hello, my name is Alef Leulekal. Um, for this summer, I had the incredible privilege of working in Kalume lab under the mentorship of Dr. Jeffrey Mensah. And my project focused on the targeting efficiency of a novel gene therapy for Leigh syndrome-related epilepsy in mouse model. Now, what is Leigh syndrome? Leigh syndrome is a rare but severe form of childhood onset mitochondrial disease. It's a disease with a very high mortality rate, with infants that are diagnosed with it not making it, or only 20% of them making it to adulthood. And it has an estimated incidence of 1 in 40,000 birds. Leigh syndrome is linked to over 75 genes of both nuclear and mitochondrial genomes. One of these genes is NDES4, which plays an important role in the stability of complex 1. Complex 1 being part of the electric transport chain in the mitochondria and plays a role in energy production. Clinically, Leigh syndrome is characterized by symmetric necrotizing lesions of the subthalamic region, basal ganglia, and brainstem. As you can see here on the MRI, these are these regions are hallmarks of Leigh syndrome. There are no available effective and safe treatment options for this disease. Since Leigh syndrome is a mitochondrial disease, it presents itself in numerous ways, both non-neurological and neurological. Past research has showed that neurological is the more important as it has a bigger effect on patients. And more research was done that showed that epilepsy is a very dominant phenotype of this disease. Past research in the Kalume lab has shown that mouse that have this NDES4 gene missing are a very clinically relevant model of Leigh syndrome, since they exhibit a lot of the same symptoms that Leigh syndrome patients have, especially seizures. More research was done to figure out where exactly this problem comes from, and it was found out that when mice with the genetic deficiency in inhibitory neurons or GABAergic neurons alone had it, they had epilepsy, as you can see here. The EEG, mice with this specific NDES4 knockout genotype in their GABA neurons had spontaneous myoclonic and tonic-clonic seizures. They also were susceptible to thermally induced seizures. So this shifted my aim to study the effectiveness of our therapeutic adeno-associated virus 9 vectors in targeting inhibitory neurons, which are the site of the disease-causing mutation in our mouse model of Leigh syndrome-related epilepsy. My hypothesis was that efficient delivery of replacement human NDES4 gene in inhibitory neurons will lead to better therapeutic outcome. And this was the construct for the AAV9 vector. So my method consisted of first intracerebroventricular injections of mice postnatal 1 to 3. And then at P60, these mice were perfused and their brains were removed and stored in 30% sucrose. Then those brains were taken and sliced into sagittal and coronal sections in a cryostat. Then those were mounted onto slides to prepare them for immunohistochemistry staining with antibodies. And then we used confocal microscope to do imaging. Now, we found out a lot of results. First, there was past research done on the phenotype of epilepsy when it came to these endosperm knockout mice. My mentor Jeffrey found out that there was significant improvement in seizure rescue when these mice have gotten this treatment. As you can see here, the ones with the treatment were able to withstand thermally induced seizures for up to 42 degrees, where only a few of them actually got seizures, while the control mice that didn't have the treatment or had the treatment without the human endospore gene replacement all started having seizures by the time the temperature increased to 42 degrees. And these were my results. As you can see up here, the first two images showed TD tomato labeling. TD tomato shows the expression of AI14 markers, which are present in inhibitory neurons. So this was what we expected. We wanted to see the presence of inhibitory neurons in both the control and ICV HNDOs treated mice. What was different was when we did the HA labeling. We could see that only the treated mice had this. The labeling of HA means that the transgene is expressing. So this showed that there was success with the gene replacement and that the mice were expressing this protein. And this is backed up by the phenotype proof that my mentor did. So my conclusion is that preclinical human endosphore gene replacement therapy in Leigh syndrome-related epilepsy is very promising, and that there is evidence of expression of human endosphore protein in GABAergic neurons after ICV treatment. And there were a lot of ways we can take this data, and we can use it to evaluate human endosphore biodistribution profile in the brain and determine the correlation between biodistribution and treatment efficacy. I would like to thank everyone who has supported me in this program. Your mentorship and encouragement has made this summer an incredible experience. Dr. Ellen Bogan, Julie, without you, I wouldn't be here. So I would like to thank you so much for this opportunity. My PI, Frank Kalume, and my mentor, Jeffrey, you also helped me a lot throughout this entire internship. So I would like to thank you as well.